Hello, everyone. So here I will be actually answering a couple of questions, mostly concerning the cytoskeleton, the 9 plus 2 arrangements, the flagella, and the cell movements that you guys have been asking about, and especially also the movement of vesicles. So when you guys take a cell and you got, let's say you open it in half and you look inside the cell, you're gonna see basically these kind of massive protein fiber networks that literally look like bridges all over the cell. Well, that's your cytoskeleton. These are literally, again, network of protein fibers that are found in all eukaryotic cells. And these are super important because they help, of course, give shape of the cell, but also keep organelles in fixed locations and also helps transport of cargo, especially like vesicles or organelles or proteins from one place to the other in the cell. So they kind of act like bridges. And they also help the cell kind of move. They have a lot of important functions that we're going to go deep inside of them. But one thing that we know from this cytoskeleton is that it's a very dynamic system, meaning that these kind of networks of protein fibers, they're constantly being formed and disassembled, meaning that, let's say one filaments or these kind of fibers are being created, they will basically be destroyed within, let's say, 2 or 3 minutes, and then they will be actually formed again 10 minutes later, and then be broken down and formed again. So that's what they mean by dynamic system. And in eukaryotic cells, there's three main types of cytoskeletons: the microfilaments, the microtubules, and the intermediate filaments. So let's start off with the microfilaments. The microfilaments are literally these filaments that are made actually of repeating units of actin. So actin proteins are basically always connected to each other to create one actin filament, and then basically two of them always come together. So they intertwine, so basically meaning they are crossing each other, as you can see here, creating this nice little alpha helical structure. And that's basically an actin filament. It's two protein chains of actin together. And these are the smallest basically type of cytoskeleton you have. And these guys are very, very important in cell movement, but it's specifically a very specific type of cell movement that we call like contraction, crawling, or pinching. And this is very similar to how a baby actually crawls. So as you've seen babies, they will put their hand forward and they'll use that hand that's extended forward to kind of, push their body forward. That's crawling. That's the crawling motion. Well, cells can do that because what happens is that with actin will kind of push a bit of the basically the cell membrane forward, kind of create this hand from the cell membrane to push forward, and that extended version of the cytoplasm of the cell membrane that's out is gonna be used then to kind of push the cell forward, so the body forward. So that's what they're important with. Then you have microtubules. Microtubules are the biggest cytoskeleton, and they're actually made of two different subunits, the alpha and the beta tubulin subunits. And they're actually repeating, meaning that you always have alpha, then beta, and they're repeating. So here and here, as like microfilaments, there's two protein chains. Here it's 13 protein chains that create one microtubule. So one microtubule is made of 13 protein chains, and the protein chains are made of alpha and beta tubulin subunits. And microtubules have a very, very important um, function, and they help to kind of transport cargo from one place to the other. This could be vesicles, this could be organelles. They help them try and be transported from one place to the other. So they kind of form in some sort of way the bridges. They're literally bridges that help things being transported within the cell. But they're also very important also in cell movement, and we'll be talking about that. But the difference between the microtubule movement and the microfilament movement is that microfilaments help cells move in a crawling manner. Microtubules very differently, which we will talk a tiny bit specifically with the with the flagella. And then you have the final one, which is intermediate filaments. Intermediate filaments are actually, as the name says, is intermediate, meaning that they're in the middle, meaning that their size is they're bigger than microfilaments but smaller than microtubules. And these guys are very different to microfilaments and microtubules because they're made of different types of proteins, not just like microfilaments are only made of actin or microtubules of alpha and beta tubulin. These can be made of different proteins. and these are quite tough. And because they're tough, they actually help a lot in the structure of cells. They're very important to give cells structure. And intermediate filaments are usually not broken down. So remember here when I was saying that cytoskeleton is dynamic, well, I was referring to microfilaments and microtubules. These guys are always being, constantly formed and destroyed. While intermediate filaments are usually just, once they're created, they always stay there. So we're going to go much more in detail between actin filaments and microtubules. And let's go more deeper into microtubules. So microtubules, where they're formed is actually in the centrosome. So centrosomes are the area where microtubules are being formed. And in the centrosomes, you have basically centrioles, two centrioles, which literally look like these barrel structures. And they're always crossing each other, and they basically come in pair. So there's basically the centrosome has two centrioles, and they're always crossing each other, as you can see here. And these are basically made of microtubules. And they're basically made of a microtubule triplets. So you have three microtubules coming together, and there's nine of them. So you have nine microtubule triplets that form one centriole. So there's two of these centrioles, and each centriole is made of nine microtubule triplets. And around the centrioles, you have something called the pericentriole material. Pericentrial material is literally basically alpha and beta microtubulin. So there's a lot of these alpha beta microtubulins subunits around. So which makes sense because if these guys are the ones that are going to be creating the microtubule, they need the subunit to do so, and that's the pericentrial material. So they're going to be picking up these alpha and beta tubulins and create these microtubule filaments. So those are the centrioles. So as we said, centrioles are very important in transporting um, cargo. They're very important in transporting, let's say, cargo from one place to the other. And I'll give you here an example of dionine. So dionine is a molecular motor. So as the name says, it's literally a kind of a car. Think about it as a car, a molecular motor, which connects to a vesicle through this dynein-actin complex. So it's this basically kind of complex that helps dynein connect to the vesicle. So the dynein will only walk from basically the cell membrane towards the nucleus. So always towards the nucleus. So if you have, let's say, a vesicle that's near the nucleus and you want to transport it, sorry, a vesicle near the cell membrane and you want to transport it to the nucleus, the dynein will do that. And it will do that through using ATP. So these are molecular motors. These molecular motors do not work without ATP. They need ATP to walk. They need energy. So that's one type of molecular motor. A second type of molecular motor is called kinesin. So I'm going to show you an example here. Yep, this is it. So this is the basically dynein, and dynein only walks, so it only goes this direction, so towards the nucleus. So kinesin is basically another molecular motor that only walks towards the cell membrane. So depending where the vesicle needs to be transported depends on what molecular motor. If it's towards the nucleus, dynein. If it's towards the cell membrane, kinesin. And these guys really require ATP to move. So this is how basically, cargo is being moved within the cell. So the microtubules literally form these kind of roads, and then you have these molecular motors like dynein and kinesin that are walking in different directions, and you have ATP. ATP is really required to push them. So now let's go talk about cell movement. Cell movement, as we said, anything that resembles crawling is all done with actin microfilaments. Actin will literally push the cell membrane forward and help kind of push the whole cell body forward, just like a baby. So that's one type of cell movement, and which is basically crawling, and that's only actin microfilaments. However, we have something else, which is basically, another type of movement that requires a flagella. And you see this actually not in all eukaryotic cells. Nowadays, not very few eukaryotic cells have a flagella. Humans, we don't have it at all, and most animal cells don't have it. It's very actually certain archaea that actually have it. Not archaea, sorry, some other eukaryotic organisms that have it. But these flagella are actually made of microtubules, and they have this 9+2 arrangement. So some people, some of you guys are actually quite, confused by what this 9+2 arrangement means. This 9+2 9+2 arrangement literally means the how the microtubules are arranged. So 9+2, so let's start with the 9. 9 means basically there is 9 doublets of microtubules. So you have a doublet that makes one, and there's 9 of them. So that's the 9. And plus 2 is that there's two microtubules in the middle. That's what it basically means. So it literally means how the microtubules are assembled to create the flagella. So the flagella is made of microtubules, and they're assembled in this kind of form. 9 plus 2. So 9 doublets of microtubules and two single microtubules. So remember, outside is doublets. So there's 9 doublets, and then here there's two singles. Now that so basically, so it's two singles, not doublets. The doublets are outside, and there's 9 of them, and inside there is basically two single. That's the flagella. So basically, here you have basically the base of the flagella, and that actually has very similar to the centriole that has this kind of triplets, and there are 9 of them. But that's just the base of the flagella. The main body of the flagella is made of this 9+2 structure. And the flagella in eukaryotes very different to the bacteria. The bacteria one propels, meaning that it actually has this kind of clockwise or anti-clockwise movement, kind of moves in circles. Well, this one moves basically, like this. So basically, back and forth, back and forth, back and forth. It doesn't kind of go in circles. No, it literally goes like this, like this kind of movement. And as I said, that eukaryotes, most eukaryotes don't have flagella. But we have this kind of structures, and these are called cilia. Cilia are these small little hairs that you actually find in your mucus sites. So, like your esophagus, your throat, your intestine, your basically your stomach. These small little hairs that basically you need to go. They what they do is they kind of first of all kind of protect and remove bacteria, but also remove kind of debris. So any kind of dead cells, they help them kind of clear it out, and they actually have this kind of structure 9+2. Those are cilia, which are literally these kind of small hair-like particles that you see basically on cells, and they help kind of the movement in that kind of way. So you have two types of movements. You have the crawling movement that's done with actin, and you have the flagella movement or the cilia movement, which basically go back and forth, and that's all because of the microtubules.